Good afternoon, everyone, and today we are diving in to another fascinating area on brain biology and beyond. We're going to be looking at the relationship between the gut microbiome and its profound connection to the human brain, often referred to as the gut brain axis. By the end of this session, we will be able to outline what the gut microbiome is and explore its crucial importance in both health and disease, looking at the main factors that influence it, its composition, and finally discuss how we might target the gut microbiome as a therapeutic intervention for both physical and mental health. As biologists, we often look at the human body through a lens of compartmentalization. We learn that they were made of trillions of cells, grouped into specific tissues, organs, and organ systems, each with their own spoke functions. However, no organ exists as an island. There is a massive amount of interconnectedness and continuous communication between our physiological systems. In recent years, this idea has expanded into something even more mind-blowing. We're not just relying on our own cells to function. We exist in a state of deep symbiosis with microscopic organisms living right inside us that outnumber our human cells trillions to one. Though we cannot see them, our overall physical health and our mental well-being are heavily reliant on a community, the gut microbiome. To guide our exploration today, we will first be looking at the biological architecture of the gut-brain axis. Then we'll be examining how the gut and the brain communicate bidirectionally, and from there we will address how this communication influences systemic diseases and mental states like anxiety and depression. Lastly, we'll be looking at how we can manipulate the gut microbiome as a groundbreaking avenue for medical treatment. So let's start with the basics of the nervous system. We know that the central nervous system, or CNS, consists of the brain and spinal cord, sending nerve innervations throughout the body. Mirroring this is the peripheral nervous system, or PNS, which weaves through our organs to create a two-way bidirectional highway of communication. The key takeaway here is that these systems are absolutely not isolated from each other. The absolute superhighway of this axis is the vagus nerve. The vagus nerve directly innervates the gut, the liver, and the spleen, playing a massive role in regulating internal homeostasis. Because this communication flows both ways, it allows the brain to subconsciously coordinate gut-related processes like peristalsis, while allowing the gut to send real-time physiological updates back up to the brain. But this connection is even more direct than general nerve endings. Let's talk about neuropods, which are highlighted in key research by Borges and colleagues in 2020. Neuropods are specialized sensory cells within the lining of the gastrointestinal tract that act like taste buds for your gut. When you eat, these cells literally taste the food and rapidly signal the brain. For example, when you consume sweet foods, neuropods signal the brain to trigger the release of dopamine, a reward chemical. Interestingly, studies show that when these neuropod cells are silenced, the active seeking behavior for desirable foods drops significantly. Research by Buchanan et al. In 2022 even found that neuropods can help the brain distinguish the caloric value of natural sugar and artificial sweetness. So where do bacteria come into this? So we've established that the brain and the gastrointestinal tract are in constant neurological conversation, but where do bacteria fit into this? It turns out that the entire neurotransmission and your subsequent response can be heavily altered by external factors, localized inflammation, short-chain fatty acids, and hormones. And that what regulates all these factors? The massive population of microbes living in your gut. They act as the ultimate traffic controller for this entire communication network. So what exactly is the gut microbiome? It is the complex community of these trillions of bacteria, fungi, viruses, and protists living alongside and within our gastrointestinal tract. To put it to scale in perspective, it weighs roughly 3 to 5 pounds, which is similar to the weight of the human brain itself. It is entirely essential to maintain existence. Mammals have never existed without them, and its role in disease and mental health has become one of the hottest, most heavily researched topics in modern medicine. Where does it come from, and where do we get these microbes? Well, the first 3 years of human life, like many things, are absolutely critical for establishing a healthy microbiome consisting exactly and lining up with critical periods of brain development. Interestingly, we acquire these microbes through various different environments, including the postnatal environment, which has a big influencing factor. There is a great difference between being born vaginally and being born through C-section that can alter our microbial diversity. Other factors play a role too, such as breastfeeding versus formula milk, whether you grew up with pets like dogs or if you spent early life in a highly sterile environment like a neonatal intensive care unit. While pet ownership and breastfeeding significantly boost microbiome diversity, prolonged time in ultra-sterile hospital settings understandably lowers it and is a risk factor for some neurodevelopmental diseases. The gut-brain axis and the dietary culture also plays a cross-generational role. Once established, a microbiome adapts to specific environmental stimuli, namely what we eat. The modern westernized diet is incredibly problematic with high ultra-processed foods and simple carbohydrates that require very little enzyme effort from our microscopes, and it drastically lacks dietary fiber and complex carbs. Multi-generational studies and mouse studies have shown that when a poor diet is maintained over 4 generations, it completely remodels the microbiome into a heavily depleted state or dysbiotic state, showing a profound interaction between our genome and our microbiome. This cross-generational depletion of our internal ecosystem may provide a missing link explaining why mental illness has risen drastically alongside the decline of our modern diet. Another major disruptor is antibiotics. When Alexander Fleming discovered penicillin, it was hailed as a medical miracle and it has undoubtedly saved millions of lives. However, because we now understand the sheer importance of the microbiome and the looming threat of antibiotic resistance, healthcare practitioners no longer hand them out like penny sweets. Both animal and human trials reveal that heavy antibiotic use in early life leaves deep detrimental downstream consequences on our health. Let's look at the hard evidence, much of which comes from animal models. A pivotal study by Des Bonnet et al. In 2015 found that wiping out the gut microbiome of mice using antibiotics led to direct increases in anxiety behaviors, a drop in BDNF, the neuroplasticity chemical involved in neuroplasticity and neurogenesis within the hippocampus, and also lower oxytocin levels, collectively resulting in distinct cognitive deficits. Interestingly, oxytocin is also found defective within autism spectrum disorder, perhaps establishing a potential link between the two. Furthermore, research by Leclerc et al. In 2017 showed that these microbial alterations actually compromises the integrity of the blood-brain barrier and ramped up systemic inflammatory cytokines. This brings us on to another major theme, inflammation, which you can watch in another video that I've recently posted. Now, the roles of the gut microbiome are many. It has a role in neuronal development, a key regulator of the immune system and inflammation. It assists heavily in digestion and produces essential vitamins and short-chain fatty acids. These microbial metabolites can actually cross into the brain, across the blood-brain barrier, and directly regulate the mood and neuronal development. Because the establishment of the gut microbiome mirrors early brain development, the two are fundamentally linked. For instance, the short-chain fatty acid butyrate can regulate genes responsible for prefrontal cortex activation. Conversely, when mice are raised in completely sterile, germ-free environments, their neurons show downregulation of over 300 genes, severely altering their normal brain development, and this is heavily implicated and linked to the microbiome disruption of the sterile environment, again found within models of autism spectrum disorder. So let's do a little pit stop of what we've covered so far. So we know the microbiome is established early in life, and we know that it's deeply sensitive to external factors like birth method, diet, and antibiotics, and we know that damaging it directly harms neural development and cognitive function. Now, let's pivot and deep dive into the exact physiological mechanisms of how these microbes alter our daily moods and mental states. So how can the gut microbiome contribute to our mood? There are 3 primary pathways through which the microbiome dictates our emotional and psychological states. This is through the regulation of systemic inflammation, the alteration of our physiological stress response, and the direct production and modulation of neurotransmitters. Now, it shouldn't come as a surprise that when the microbial community falls out of balance, a state known as dysbiosis, our health quickly deteriorates. To put it simply, gut microbes and their metabolites tightly regulate the permeability of our intestinal walls. When the microbiome is healthy, the barrier is secure, but in a state of dysbiosis, the gut lining becomes leaky. Microbes and toxins that are supposed to be trapped inside the gut escape into the systemic blood circulation, and this can trigger a massive inflammatory cascade, spiking pro inflammatory cytokines and microglial activity throughout the body and the brain. They do this through several mechanisms, including lipopolysaccharides within the cell walls, activating the immune cells, short chain fatty acid synthesis, C reactive protein release as well. Now let's have a little closer look at these mechanisms. Lipopolysaccharides, or LPS, are components of the cell wall of certain bacteria, and when these bacteria overproliferate, often driven by high-fat, high-processed diets, they degrade the integrity of the gut epithelium, causing a leaky gut. And this circulating lipopolysaccharide causes severe insulin resistance and chronic inflammation, closely linking metabolic disorders like type 2 diabetes and obesity into systemic inflammation. On the positive, short chain fatty acids regulate these cascades by modifying the NF kappa beta pathway, which is a master control of the inflammatory response. For example, a beneficial bacterium called fecal bacterium progenitorly actively boosts butyrate levels, keeping inflammatory marker levels low. This bacteria is severely depleted in individuals with obesity, and this may provide an explanation for why obesity, chronic inflammation, and poor mental health are so frequently linked. Next, let's talk about stress. Our primary stress pathway is the HPA axis, the hypothalamus pituitary adrenal axis. In response to danger or anxiety, it releases cortisol to increase alertness. This is an incredibly important evolutionary survival mechanism, but when it is chronically activated, the long-term health consequences are devastating. Remarkably, the gut microbiome directly regulates the sensitivity of the stress response, potentially through affecting glucocorticoid receptor sensitivity, amongst other mechanisms. In germ-free mice models with no bacteria, the HPA axis is hyper-reactive and goes into overdrive under stress. However, researchers found that stress hyperactivity could be entirely reversed by simply colonizing their guts with bifidobacteria. Furthermore, fecal transplants from stressed human individuals have been shown to directly induce depressive behaviors in rats, demonstrating that a stress microbiome can physically transmit anxiety. Neurotransmitters represent the third pathway, and our microbes possess highly diverse metabolic pathways capable of synthesizing or altering chemical messages that talk to the brain via the vagus nerve. Take serotonin, for example, the chemical responsible for mood stabilization, or at least part. A staggering 90 to 95% of the body, serotonin, is manufactured directly in the gut, stimulated specifically by certain bacteria. It also is involved within the synthesis of dopamine, which is out of balance within disorders such as ADHD and schizophrenia. So, what can we actually do to improve our gut microbiome? Given everything that we've discussed, the ultimate question is what can we do to improve our gut microbiome? Apologies. Given improve our gut microbiome and protect our physical and mental health. Today, we'll talk about four main levers we can pull: increasing fermented food and fiber, reducing alcohol intake, eliminating artificial sweetness, and exploring advanced clinical options like fecal matter transplants. Let's look at fermented foods. Dr. Justin and Erica Sonnenberg at the Stanford University conducted incredible studies looking at how dietary interventions affected our intestinal ecosystem. Originally, scientists hypothesized that simply ramping up dietary fiber would yield the greatest microbial diversity and best reduce inflammation. However, the actual clinical results were surprising. While high fiber diets were beneficial, it was the consumption of fermented foods like kefir, yogurt, kimchi, and sauerkraut that dramatically and rapidly increased overall gut microbiome diversity and directly slashed systemic inflammatory markers across the board. When it comes to alcohol and the gut microbiome, we should be reducing our consumption of this. Historically, alcohol has been celebrated for its powerful antiseptic properties, And it's what we have used in the past and still do in occasions to sterilise medical wounds. We rub it on our hands via sanitizer. However, when we consume it, it acts as a sterilising agent inside our digestive tract, devastating microbial diversity. Clinical studies of individuals with chronic alcohol use reveal a severely depleted microbiome. Specifically, alcohol severely decimates beneficial anti-inflammatory bacteria like the butyrate-producing clostridiols, which allow highly pathogenic pro-inflammatory bacteria to multiply and trigger systemic inflammation. Sweeteners, which have grown massively within the Western diet in recent years, are also another silent disruptor of the gut microbiome. Animal and human trials have raised major red flags regarding their safety. In a controlled study by Palmas et al., rats given aspartame in their drinking water for just 8 weeks showed a significant rise in pathogenic gut bacteria and developed distinct insulin resistance. Similarly, research by Nettleton et al. In 2020 confirmed that artificial sweeteners may cause a sharp spike in inflammatory strains of Clostridium, completely altering metabolic homeostasis, which can have a downstream effect within various different neurological disorders. Studies in humans have found mixed results, um, with some finding no difference at all after taking sweeteners, while others finding significant changes. Finally, let's look at a cutting edge frontier of medical intervention that is extremely gross sounding but incredibly promising, and that is fecal microbiota transplants, or FMT. A fascinating clinical case report by Dole et al. In 2022 looked at using FMT to treat patients suffering from major depressive disorder. They took healthy donor microbiomes and transplanted them into depressed patients. For the first 4 weeks, the results were incredibly promising, with patients showing a drastic reduction in depressive symptoms. However, the study highlights the limitations of current practices. In one patient, their depression scores dropped from a severe 21 down, by a significant 21 points, back down to 9 at week 4, but climbed right back up to 19 by week 8, suggesting the transplant alone wasn't a permanent cure without altering other baseline lifestyle factors. Conversely, the second patient experienced sustained long-term relief, with their scores dropping from 31 down to 10 and staying low 8 weeks later. This shows that fecal matter transplants have massive therapeutic potential, although more large-scale research is necessary. To close our session, if you would like to explore this topic further, I highly recommend the following books: Dark Matter, which covers the science of the gut microbiome in more detail, by Dr. James Kinross, and the book Good Guts, although I can't remember the name of the authors. Thank you very much, and if you've got any questions, I'm happy to take them. Do check out the rest of the videos. Thank you very much.